Ready? Okay. Oh, where is your friend? Oh, she don't like it. Okay, let's start with the Rift Valley fever. Okay. Okay, so this is mostly the disease happened in Africa, but I'll tell you the context that there is potential of coming that virus to India too. So that's why I included this in my talk. Um, Here you can see that a lot of countries, Kenya, Tanzania, Somalia, so all those from African countries, and starting from 1930s to still so far 2020, 21, 22. I think it's reported 2024. So there are a lot. So it's a color coded, right? Blue color and I should say pink color. So blue colors means it is mostly in the human and animals it has been outbreak detected. And when it is pink color, it's mostly animals because that Rift Valley is affect both animals as well as humans. So you can see mostly in Kenya, you can see major outbreak happen, and also South Africa, but others like sporadic incidents happen that virus. So again, like the previous virus, Anton virus, because those both are, I think, Buniavirials, and it's a family new viridae and flavivirus. Again, this virus we also covered in our review article. I'll tell you. So there are three segments. Last time also I told there are in like a Hunton virus there are three. Here also like large, medium, and small. There are polymers, nucleoproteins, which is called large segment, medium segment, and small segments. And each segment has the different proteins they give. Like in medium segments, there are non-structural proteins, but also produces two important glycoproteins, GN and GC. The small segment is mostly non-structural protein, but also have a nucleocapsids, and it is also L segment is mostly L protein, which is also non-structural. And there are other non-structural proteins. So this is basically the structure, like a schematic diagram of those viruses. Again, the replication, nothing to talk. Start working. Yeah.
Yes.
Sorry. So there is nothing to talk more about the replications. It's similar, like we mentioned, goes from encapsulated. After virus attachment, it gets encapsulated, and then it gives like cDNA, complementary RNA. Sorry, so complementary RNA, not DNA. Complementary RNA, and then make lot of viral RNA negative strands copies. And ER is more important for the production of non-structural proteins. And then the assembly happens in the Golgi, which is kind of packaging unit, and then the virus is released from the cells. So this is important how the virus transmission happens. The major culprit here is this in this mosquito, Aedes amphilox, those species. This is the more important for the transmissions, and there are another is like sylvatic cycles, which is within wildlife. It is always present within the animals. The people have studied already, goats, camels. Maybe I'll talk some of them animals. These presentations, and they are vertical transmission. You know what is vertical transmission is, right? Vertical transmission and horizontal transmission. There are two kind of transmissions. I think it should be very clear. Horizontal means from like I am giving virus to Dr. Reddy. How? Maybe I'm coughing, or I'm sneezing, or I touch something, I'm infected, I'm touched something, and I do handshaking, and he got it. So this kind of a horizontal transmission. Vertical transmission is like pregnant mothers infected, and the virus transmitted through the fetus, through the uterus, cord blood, and those. So that is called vertical transmissions. So those two transmission is always the horizontal transmission and vertical transmission. So vertical transmission is always there. Like if the zero for goat are pregnant, they pass that virus to the babies, and same for humans too. They transmit the same way, and also the contacts happens multiple ways. Not only mosquito bites, but also you have a good farm. You have a lot of animals. You have goats, sheep, camels, and you just pet them all the time. But you never know. You never tested those your pet animals. They are infected already. Those viruses or not, and you get from them directly through contact with tissues or fluids, those things. So that's the major things the virus transmission happens. And again, it definitely, mosquito. We all knows malaria. It's very prone. Uh, uh, we need rain. We need a stagnant water that the mosquito can breathe, right? That's a fundamental question. I mean, so that's always there. So and there is another, like this virus is also, if you general question, like why mosquito borne is more prone to all countries, like mostly developing countries, right? The reason is no. The reason is we don't take care of the society. We are not clean enough. Why the developed country it's not get that kind of mosquito? I mean, West Nile, except that. Um, but their mortality is not that high. But if you see malaria, dengue, chikungunya, lot of death and lot of happening. But because the most thing is like we are not proactive enough to take care of. Okay, my backyard there's pile of water there and mosquitoes breeding. I don't take care of it because it's a neighbor's place. I don't care. But you have to be careful, right? Yeah, that's why I say very proactively you have to work on, or maybe talk to your neighbor, say, hey, there is a breeding ground of a mosquito. If I get sick, are you going to pay my health insurance? So that's the way talk should start from there, right? So I think people should be more aware about it, and that's happening because in India and those countries, a lot of rainfalls happen, a lot of humidity, and those are the breeding ground for those mosquitoes. America, cold, mosquito die. That's why I talked about West Nile yesterday. Like it's seasonal, right? When there is summer, there is more rainfall. You see the peaks, but winter you don't see because all frozen. Okay. uh, so that's the another thing, but India never snows, and if it snows, unfortunately, it's a big celebration, right? But it's a lot of rainfall, lot of humidity, so we should be careful those things. I mean, that's the thing, if we do allow our public service in that way, which is unpaid public service, right? Take care of our drains system, our backyard, our neighbor's place where there's more water, accumulating or breeding ground is generating. If we can take care of those, I think we can cure a lot, especially mosquito-driven diseases. So humans is self-limiting febrile illness. It's not too much. I mean, because it may have a mutations. It's still at present. It's like that. Self-limiting febrile illness, 1 to 2% more severe disease. 2 to 6 days incubation period. Anybody can say what is incubation period is? What that means? I talked briefly yesterday. Huh? What? Incubation period. What do you mean incubation? Like, baby is in the womb of a pregnant mother. They're incubator, right? The uterus is an incubator. And the baby is born, going there for 9 months, 10 days, right? Theoretically. I'm not talking early baby or late baby, but that is so that you call incubation, means baby is sitting there without any symptoms, and once it come out, it's come out, right? So same like virus, incubation means the virus is not showing anything. You got the infection, maybe I got in plane on certain Sunday, but the virus is incubating now. Maybe Friday I'll find out I have fever and all those kind of headache and all this. So from Sunday to Friday, that is the incubation. So I have no clue what is happening. But on Saturday or Sunday, when I find out, oh, I have a fever, so that means for these maybe 2 to 6 days. Some people have early, some people have late, based on our immune system.
It's not like critical amount or number has to be reached, then only can analyze it's that.
What do you mean?
No, incubation period means kind of like it reaches a critical mass or number, say for example number of viruses body.
Oh, sure, yeah.
Then it
starts showing the symptoms. Yeah, but it also depends. Like my immune system or her immune system or yours is a different, right? Because we are all human population are heterogeneous. Until the twin babies, they are mostly homogeneous. Like there's twins have similar kind of thing. But so my immune system may be so bad like than yours, but you may takes 8 days. I may show up my 3 days because my immune cannot catch up that way to control, and my virus replicates so high within 3 days, and I show symptoms. But yours takes little time.
So does the infection always leads to the symptoms of disease?
Symptoms and disease are together, right? Symptoms is the outcome of the disease. Yeah. So when you saw that I have a fever or brain fogginess or I have a headache and tremors, you have a disease. Those are showing the symptoms.
What I mean is that like you get infected, but the amount of the infection, maybe say number of the numbers of the virus that got into the body in the beginning. So if they are not sufficiently enough or cleared by the immune system, then there wouldn't be any disease there.
Yeah. That's why sometimes dengue we discussed, like some has no symptoms, right? So that means you have not got enough virus, and your cell is able to get this and eliminate it. So macrophage comes first. Okay, we have a very good system in our body, like AK-47, right? The macrophage, neutrophils, they come first, and they say, oh, this is a foreign thing, and they just grab it by phagocytosis. That's the mechanism they call phagocytosis. They phagocytose, they eat, and then they kill it. So as Dr. Reddy says, is perfectly correct. Like if you have not enough viruses and the body can tackle it, you have no symptoms. But I got it, but my body cured it. So it is kind of unknown, no report, nothing. That says that's happened. But if I got so much, I got 10 mosquito bites last night, and all are West Nile, you're definitely getting something, right? So you have a huge load of viruses caught in your body. Okay, so incubation period. So I think you're clear right now. I hope. Some patients experience biphasic febrile illness. Means there are a lot of studies like biphasic, like you come, fever, it go down. Again after a couple of days, the fever comes and go down. So it happens also sometimes. The hepatitis, jaundice, hemorrhagic disease, ocular disease, photophobia, reduced visions, blindness. So it all kind of related to this. Like so much light irritate you. You cannot see, blind spots, retinitis, inflammations in the retina, and you can see the eye has the hemorrhagic spots on those things. Abortion, miscarriage, because it's a vertical transmissions. So sometimes pregnant mothers, if they get it, there is abortion of the babies. And this is kind of like how many percentage of cases you can see, like when it is with HIV, it increases drastically because already the patients are immunosuppressed, right? It kills those CD4 cells, which is Another way, CD4 cells called also T helper cells. So, keep in your brain, like CD4 means it's a helper. What do you mean helper? Means, okay, you helped me to give this bottle of water, right, to drink. So I'm drinking, but you helped me. So same like CD4 helped. To whom it helped? CD8 cells and B cells. Okay, what they do? CD8 cells makes anti-cytokine productions, then anti-granulocyte function, killing function. So CD8's function is those. But CD8 cannot do by himself. It needs a helper. I cannot get the water until you give me the bottle. So you are a helper, and I am the like CD8 cells, right? Same like CD4, help is needed for production of antibodies. B cell itself cannot do it. CD4 is needed. So that's why what happened in HIV, we don't get enough antibody, we don't get enough CD8 response. The reason is the helper, the person who is going to give me water or give me those things, is no more. If those people are not there, I cannot present. They are the helper for the computer. So the same thing like CD4 is very important for any functions. Keep in your mind, the CD4 is called T helper cells. CD8 called T cytotoxic cells, means it kills, and B cells is the producing antibody. Okay, so B cell has a different pathways. It calls plasma blast and then plasma cells, which is giving long-term antibody production. Once it generates plasma cells, it goes to the bone marrow and it stays there for years till we are die, and it producing antibody. So that's a basic fundamental question. So what happened, like the reason I'm saying, like when a HIV positive is there, the cases increase. The reason is because there is no CD4 response there to generate quick antibody, quick CD8 responses. Stuck. Okay. No. Okay, so in animals, yeah, so the animals you can see also there are a lot of similar lesions because it's also present in the sylvatic cycles where the animals also get infected. In sheep, the same thing. Just to not to remember, but just to go through it like 24, 36 hours of incubation period, and the same kind of symptoms like fever, loss of appetite. Those animals has goat also similar symptoms, but do not always experience febrile. So some species difference there. Cattle less susceptible than sheep. Camels less susceptible than cattle with asymptomatic infection. So they got infected, but they don't show off often. They have abortions, ocular discharge, those things. But animals, right? We don't care too much. Even if you have a pet, we really care about. But by looking at one animals once, oh, I don't think any changes happening, right? But humans, you got that part. So aerosol exposure. So what they did, like African green monkeys, so which is also another good model to study those disease. So they gave aerosol exposure, and then they looked over different days, and they looked over different parameters of the immune system. So what is happening, because that is always important, like an animal model, right? Why you do animal model? To mimic the disease, what is happening to human. Because if I come here, I may say, okay, I got infection when I'm traveling, because I'm telling you, right? But there is no proof. I may got from USA also, and it takes longer incubation time. Maybe I'm showing the symptoms, right? So that's why people make an animal model, so where you can infect the animal and observe longer time, say what is happening. So many studies, when if you are interested to do the biological science and like molecular medicines or anything, when you're doing experiments, you have to design the experiments. Okay, I took an animal model XYZ, maybe small animal mice or rat or guinea pigs or hamster, ferrets, large animal like monkeys. You cannot do chimpanzees on gorillas now. It is totally unethical now. But large maximum you can go to monkey model, non-human primates. I think ICMR in Pune, they have monkey models. So you can do those studies. And then it goes to clinical trial. go to the human. So animal model is very important to fine tune what is happening day one, day two, day three, that point. So African green monkey, so they use that study, and they infect those monkeys, and then they measure every day. So they did different tissue collection, blood collection, and the measure. So this is maybe not so important for you, but basically you can see the caspase tree, which is a marker for apoptosis. The cell is dying, cell happening. So starting from day 2 and day 7, you can see there's caspase 3 upregulation in CD8. Means CD8 cells, which is more important for killing activity. The CD8 cells also dying. So that is another feature of this virus. And weight loss and death may happen. So, along with that, these are the cytokines I mentioned, like inflammatory cytokines, IL-8, MCP1. This is mostly produced by monocytes. Soluble CD40L, interferon alpha. This is all innate immune functions. Interferon alpha is the innate function. It is antiviral, innate immune response. So they increased. CD4, CD8 T cell proliferation also. So proliferation means once I got this infection, the cells try to control it, right? So body try to control it automatically. You have not to tell body, hey, do something. No, body cell already programmed that way. And then the CD4 and CD8 try to multiply. So proliferation means they try to make more. So if there are 10 cells, they want to make 100 cells so that they can control this infection. So that is happening here. And then you can see the later point, the granulocytosis, means the granulocyte, which is a white blood cells, there is a lysis happening, late cytokine expression happening, there's some cytokine dysregulation in the brain, and also tissue destruction, pathology, and also neurological signs. So all this thing happening, even it goes to the brain. So basically, we talked about what are the symptoms happening, like eyes inside the brain cavity, right? So it's happening in the brain. It's all the tissues, everything's dysfunction happening in the virus. Remote. Next slide, please. Okay, so the diagnosis, it's a general, what I also discussed, like molecular diagnosis. You can do lab, RT lab, or real-time RT-PCR. Also, next generation sequencing. Sequencing is another nowadays. People do to sequence the virus easily. It does not take too much time, maybe one day, and then you can do the whole sequence for the virus may take some time, but for a gene, you can easily do sequencing and then find out, okay, this is the virus, this is the species. And a lot of serology we already discussed, like virus neutralization assay, plaque reduction assays. Plaque is mostly used for, some of the virus make plaques, like, you can see like spots. We'll discuss that in day 4 or 5. And that's another assay is mostly popular. Indirect immunofluorescence we discussed. IgG, IgM antibodies, this is another assay we use for detection. And then virus isolation, this is kind of gold standard to isolate the virus and characterize that this is the virus responsible for this disease. I'm not going in details, but basically for this virus, there are so many animal models have been used, but none of the animal models is actually perfect, right? Because none of them are like a human, right? So because they are mimicking the human patients. And their immunology is like a rhesus macaques are very close to this, but again, it has no ocular disease reported. But human, we can see ocular disease. But also it needs like BSL-3 laboratory, because very few lab can handle this and that kind of studies. So you have to be careful on those because it's a BSL-3 viruses. But other animals has a lot of lacking. Some animals shows the disease, some animal does not. Some are more susceptible, less susceptible, those kind of things. Next, okay. So this is another important part that virus does is disseminated intravascular coagulation. So, like Hunter virus is also do the coagulations. Widespread activations of the coagulation cascade, like we saw there definitely needs like platelets, bradykinin, those are important blood markers or growth factors are responsible for making the coagulations. And you can see many blood clots in small blood vessels throughout the body. And so when you have a blood clots in the body, in the vein, it compromises the blood flow, right? And so it uses clotting factors and platelets, as mentioned, and it leads to hemorrhage. Similar like Ebola, Marburg, which we'll discuss tomorrow. These are BSL-4 pathogens. So Ebola, Marburg, it has a similarity of those because you can see, I might have to give some pictures on those. So you can see, like, my hand is good, but I can see there's some hemorrhage happening, skin. So that's the hemorrhagic disease. Or, people are standing, they can see the bleeding from the nose happening or from the eyes happening. That's kind of hemorrhagic disease. That's why they say. And that's why you have to be careful. You cannot touch that patient because the virus is in the blood. So that we easily can get contaminated with those. And that's happened long ago in Africa and other countries because they don't know, oh, blood is coming, let's wipe out, put the band-aid and all those. You transmitted already the virus. So that's why nowadays when it happens, especially hemorrhagic disease, even if blood coming through urine, blood coming through feces, everywhere you can see simple organs like bleeding from the ears. So you should not say, hey, what is happening. So that maybe if you touch it by bare hand on those, you are getting also contaminated. So those are very common, that kind of disease. So epidemiological, it's the same what we discussed already. There's Culex and Aedes mosquitoes. It's common in high precipitation, where it's a lot of, as you know, this is mosquito borne. So you need rainfalls. You need a lot of rainfalls, high humidity. Those are responsible for this. So who are the risk? Like farmers, veterinarians, butchers, or the living close to the livestock. They are because they are not literate enough to know, okay, this animal may have this, right? And neither anywhere, the farmers are well educated. Oh, I have a farming 2,000 cattle, but I don't know those. I think that's kind of another things like farmers should be educated on those aspects. So again, the farmer got infected, and then transmitting to their close friends and family members, and that way the transmission may happen. Those. And mosquito bites day and no person to person. So this is the interesting that no person to person transmission documented. It's mostly the picture I've shown, like it's mostly come from animals. Animal to human transmission happens very easily. Mosquito to human transmission happens very easily, but not like human to human. So that's yeah. If you touch that, if there is hemorrhage, that is the only cases. So. Preventions is there is a vaccines available for animals. So that's the good thing that you can use those vaccines, live attenuated virus as well as formalin inactivated virus, available for Rift Valley fever vaccines. But there is no vaccines available for humans. You should go to that literature. There are a lot of references, whatever so far studied. Um, for the Rift Valley fevers, for the humans, there are a lot of study going on at the preclinical levels, as well as some clinical trials has been started. But there are options there, like virus-like particles. You can use the DNA vaccines. I think DNA vaccine is very common nowadays to use for every disease, but no success yet so far for other diseases. Recombinant virus that lacks non-structural proteins like this, surface and membranes are more live attenuated virus. So you can generate those vaccines for making those vaccine against the humans. But for animals, they have vaccines, so that's good part in it. So I think that's the last for. Okay. Oh, this one I like to emphasize that there's a article came. There is an increased threat of globalization, and so this is as a 2023. There's paper. There is chance of this Rift Valley fever globalization. So as I said, those mosquitoes are prevalent, right? Asian country, China and others, due to the temperature and humidity. So Uh, you can read those articles. It's very nicely reviewed that the options that there is a potential that it may lead to other countries, and India is one of them. So that is very important, and it's a company in mosquito vectors, large livestock industry, because these are already we have in India, right? A huge livestock industry, and so there's potential to get those viruses easily transmitted to India, because Rift Valley fever is not uh, present right now, or at least I can say not reported yet. So if it is a very unrecognized, or those things are to reduce it. So those are the possibilities there. So okay, so this is the end of the talk of this Rift Valley fever. If you have any questions, please raise your hands. Yeah, I mean, for example, well, first of all, I think it's kind of ignorance, I should say, because mostly outbreak happen in Africa. So who want to spend money in Africa to study those? Yeah, if it has happened to United States, definitely we have a lot of progress on the suspect. But it's Africa first of all. So I think a demographic region of the outbreak. So people have less interest, and who will spend money to do research and make the vaccines. So that's the first thing. And second thing, we need BSL-3 lab facilities to do those. Africa does not have that capacity to do their research on those, except the South Africa, which is richer than others, rest of the Africa. So, I mean, that's the question. But there are studies on preclinical trials, different animal model they are doing, even though we cannot go directly to the human, right? We have to show the data from preclinical studies. We get approval from the FDA for the clinical trial. So that's another thing. Like, it's not that enough data yet that okay, this is the right candidate to test. So that's my take there. I mean, it's definitely lack of interest, because they call it African virus, uh, and second thing is who will pay for it. Uh, but if it has happened to any developed countries, and definitely they have much progress on those. That's why the HIV happens. Like when HIV is there, they say 1930 is it's still there in Africa, right? Nobody, people died, nobody cared. When it happened in 1981 in Los Angeles, two guys got infected. Then people think about, oh, this is very important. Why in my country it has happened? Then so much research, so much funding and everything. So it's definitely need-based research, right? Yeah, they don't care because oh, it's not affecting us, right? Why you should worry about it? Until WHO is really interested to provide more funding and other things. So. Okay, pox. I think yeah, we know that pox virus, right? Monkeypox. Now we don't say monkeypox, right? It has been nomenclated new mpox, because I don't know why they changed it, but when it started, it was monkeypox. So first couple slides I'll go very quickly. Um, Maybe not this slide. So it's a pox virus is a DNA virus, right? So far, yesterday and today we talk about mostly RNA virus, right? The pox virus is a DNA virus, and I don't know everybody has a pox vaccine for against pox virus. I don't think, because the new generation don't get pox vaccine. I got this maybe because I'm an old generation. I think you have read on this. I think yeah. I mean mostly uh, 30 above, they should have a pox vaccine. I think it's eradicated. I forgot when it's eradicated. I think 1975 or so. It has been eradicated. But before that, everybody got pox vaccines, and it was a huge endemic. It's called pandemic viruses. So much death and happened, but still now I have taken. I think I took pox vaccine when I was in child, and then when I started in UC Davis, my first postdoc, I was working with alpha vector. So it's another vector which is same like pox virus family. So as I'm working on the recombinant vector, I have to, from the CDC, they said I have to take another pox vaccine. So I took another pox vaccine that time. So my maybe antibodies titer is very high now. And then they said, even for, that's why when I moved to NIH, they also tested whether I have pox vaccines or not. But still they have like pox vaccines kept for, especially for military peoples, for like fast frontiers health workers, and especially for war. If some country has a pox virus, or because they know it's like new generation don't have the pox vaccines, variola vaccine, so there may be a bioterrorism may happen. Like people may have somebody, terrorist group, they have the virus there and they can use as a bioterrorism. And so let's say United States has the stock enough to vaccinate all their army peoples, and so that they are protected from any kind of outbreaks. But not, they are not usually used for like general civilians yet. So until this. So you can see it's a big virus, 360 nanometer. So the largest disease-causing virus is this. It's a brick and oval-shaped double-stranded DNA, and it is so huge. You can see 140 to 300 kilobase per. It's a huge virus. It's so much protein there. And recombination is also very common. Replication happens in cytoplasm, DNA-dependent RNA polymers. This is the first animal virus to be visualized. So you can see some good microscope, the pox virus. The first virus to be grown in tissue culture, titrated, purified, and chemically analyzed. This also first immunization happened, and first virus to be eradicated. So this is the virus which has been eradicated first time. So this is the electron microscopy pictures, how the virus looks like. It looks like a brick separate, you can see. This is the vaccinia virus. I think this is the ORF. This is the molluscum contagium, that is another virus. And this is the model they saw how the core protein is there, the DNA is there, and then lateral bodies. They have two lateral bodies. That's unique for those viruses. They have their own functions. So, I don't want to say it's kind of similar pattern of the replications. Cell enters through fusions, it has receptors, then protein, then uncoating happens because the envelope came out. Then it generates all non-structural proteins. The DNA goes in the nucleus, and then it's multiply or replicate. It's make different strands because it's double-stranded, and those non-structural proteins, processed by endoplasmic reticulum. Packaging happens in the Golgi, and so all the wrapping and everything happens here, then transported. Either so there are two ways of releasing the virus. Once they can do the lysis of the cells, so they kill the cells because they multiply so much and then kill the cells. Or they can endocytosis, they can release so that another new virus can infect and use that same the same cells for growing. So this is a cordyceps. So there are a lot of viruses. You can see here the more important is that orthopox virus, which is variola, which is smallpox. And another, we'll be focusing on monkeypox. And it's a similar kind of symptoms, they say, in like smallpox. And it's the primary host is rodents, but vaccinia is another model. It does not do anything too much. It's a lot of use of vaccinia virus for making vector vaccine. Vaccinia is another good vectors vaccines used for different vaccine making platforms by different companies and others. Cowpox, some lady was asking, I think you also, the cowpox, why you can use the cowpox for smallpox, but why not, take something from bad tissue and use for for the human controls. But we discussed a little bit on this. Uh, so the cowpox is another thing which is very, and that Edward Jenner has done the cowpox postulates and used for the smallpox control that time. So, but there are others also. There, you can see that's not does not cause a human disease, and we are not focusing on other animal disease here. Mostly focusing which is actually affecting the human population, but there are other viruses which mostly affecting different animal models and other things. So I'll skip those because these are animals. Uh, let's go to smallpox. So the variola virus, okay. So this is the smallpox-like disease described in India and China as early as 15,000 BC. Because the physical evidence of smallpox-like disease found in Egyptian mummies. The mummies, if you see carefully, and people have studied already, they have seen they have like pox-like, pox-like lesions in the body in the mummies. But that time, that we see, there is no kind of diagnosis that they can say, oh, this is caused by pox viruses. By the early 20th century, smallpox was endemic in most of the parts of the globe, up to 125 countries around the world. So this is kind of a progressions. They got through aerosols route. It goes aerosol and then the virus is incubating. So you can see the line where the incubation and all those time points when the disease has been, shown or the symptoms has been, find out. That's the A pictures, and the B is also showing how the virus goes in the lung, and then viremia happens, and then it comes to the skin, because it is major lesions shown in the skin. So let's discuss the top A. So virus goes in, and then the incubation for 7 to 17 days, and we talk about incubation day, what that means. And then, the virus is big, virus is big, and then we saw the symptoms. So it's a prodrome stage, which is 2 to 3 days, and then we can show the lesions in the skin. And eruptions, papules, and then vesicles, and then you can see pustules, and then it becomes like full of liquids and all those, and then it becomes crusted, and then discoloration. So it takes, when it comes, it maybe in a couple of weeks when it's come this stage. So basically, the virus goes to the lung, and then it goes to the pharynx. So those are the mucous cells, mucous membrane. So it goes to the pharynx and goes disseminated the regional lymph nodes, and then high viremia, because lymph node is a major source of all virus replications. Okay? Then the high viremia, then the virus spread in all the tissues like spleen, liver, reticuloendothelial systems. High virus, prodromal stage means high virus, and then it goes to the dermis. And that point, when you have high viral load, you are also easy to transmit others. So if you have friends or anybody, or if you have family members, if you sneeze or cough, it's easy to transmit to them. And this is the kind of intraepithelial multilocular vesicles, like a budding features in the skin. You can see. I'll show some good pictures in the next couple slides. And what happened to the immune response? Here, infected macrophage and activated T and B cells, they try to control it, but they cannot do too much. But until the adaptive immune response comes. So adaptive immune response is the T and B cell functions. Okay. So when the neutralizing antibody comes, then you can see the viral load go down. So this is the red color. You can see the viral load. So viral load goes down when in the end stage, but it is still peak level, and that is important also. Like good viral replication is also important. It's a balance, because a certain amount of immune response, a viral replication is needed to have a greater T cell B cell response. If you don't get enough viral replication, you don't get good immune response either. Um, so that is happening. And so here is the incubation time point, and you can see oropharyngeal secretion. So you can easily transmit also So this is the peak time when you have the eruption papule, when you have the lesions in the body. That is the time you can transmit easily. There are so many types because it's an old virus and it's a lot of studies has been done that time. I don't think people have followed the BSL-3 facilities that time, but people have studied. I mean, we have a lot of knowledge on those. So there are ordinary and within ordinary they have like discrete, semi-confluent and confluent, and then variola seeing eruption, which is fever, and raw rash, they're just a fever, and then modified version, and then flat version and hemorrhage. So there is one, two, three, four, five. Five different types of smallpox happens. So not everybody has the one time. I may have a hemorrhagic. Hemorrhagic happens, which is very lethal. People die of that point. But some has flat, some has variolas. So we can discuss more on this. This is the ordinary. But you say ordinary, they have like a discrete semi-confluent. So if it goes ordinary, what happened? There's sudden onset of fever, head and backache, day 3 improvements, fever down, evolution of rash, macule, papule. So these are the terminology of how the rashes happen. So you don't need to remember, but they just remember there's rashes and then secondary fever after 4 days, and then tertiary fever, bacterial superinfections. Because those rashes, if they rupture and if you don't take care of properly, if you don't have, people who stay lying in the bed or sitting on, if you don't take care of your bed sheet or don't clean it, the bacteria is there. You may easily get a superinfection with the bacterial infections, and that complicate the situation further.
Does the image show the revolution now? The revolution being shown, or is there the picture there?
No, it's a what? The evolution rest? Yeah, these are the stages. These are the stages.
Corresponding to that, no, right?
To some extent. Yeah, we are not showing all of them, but to some extent you can say this is the vesicles happen, this is the pustules. So pustules, the white thing, this is the pustules. So when bacterial infection happens, you can see that kind of whitish, those, because you got bacterial infection somehow, because let's see. So you have not taken proper precaution because bacterial goes in and you have to. So this is ordinary discrete, how the ordinary discrete, this is kind of this areas of normal skin between pustules. So you can see normal skin in between pustules. It's not all together. You can see the normal tissue and pustule came, even normal tissue, that kind of, so it's called ordinary discrete. So this is semi-confluent. You can see this is the pustule confluent on face but discrete elsewhere. So you can see this is, because it's semi-confluent, so here it is confluent, right? It's full of pustules, you can see the eyes, and that's another thing, the picture is not, I think you are all 18 above, right? So it's not a problem, but sometimes it's, that's happened. One of my family member that time, he has a pox, severe pox, smallpox, and he said it causes total deformation of your skin. People get blind also because the pustule goes into the eyes. People got blind. People got deaf also. It's inside the ears. That's why it's a terrible disease. It totally changed the structure of human being. Like people put acid in your face, that kind of thing. If you see, once it keeps marks everywhere. And that's another feature. And you see the hand. So do you think it will be normal hand anymore? No, it will be totally scarred hand. So it's a confluent rash on face and tongue. And so you see there's not too much empty space there. It's a confluent. The whole skin is confluent. And in that case, they say like a day 12 to 12 mortality happens because it's intolerable. It's so painful, and people, especially the kids, and happen, and they scratch. And if you scratch, it's forget about it, right? It's like you contaminated everything. You scratched your whole skin, you're bleeding everywhere. Then it's a mess. That's why a lot of people do that time. I read a lot of papers like they cover the whole body, so then and tie their hands also. They it's very very easy to irritate, no? To scratch the end of those things. They try to protect, they cover those fingers, wear gloves and those things, just to cover those, it should not damage further. So it's a very bad disease here. The flat form, you don't see pustule kind of thing, right? It's a flat. But that is all. This is called flat version of smallpox. Apex flat, usually fatal. So if you have a fat, this is actually very fatal disease. Um, this is hemorrhagic early. You can see the hemorrhage is happening. Some of the like red color. So the hemorrhage is already started there. So widespread hemorrhage in the skin and mucous membrane, petechial, purpuric rash, and illness. So you can see those. Earlier you saw it's like a confluent, like looks like the fluid inside, but here you see kind of black and reddish, right? So those means they are hemorrhagic. This is very bad pictures, but just to show hemorrhage into the base of the vesicle latent illness, it is happening. You can see how hemorrhage is it's totally deformed your face totally. But there's always confused with smallpox and varicella. If something happens in your, especially the kids, varicella very common in kids and also in adults who are actually taking corticosteroid and something, because those varicella is also herpes virus. Smallpox is a pox virus. I think in this room at least somebody in your family or have a varicella. Yes. So this chicken pox is very common. It's not only for the kids, but also adults get it. When adults get it, if because once varicella, it's same like HIV, it does not get rid of it. It is staying somewhere in your lymph node or somewhere. And once you compromised your immune system, compromised, you are taking corticosteroid, or you are sick some reason, you can get those symptoms. The difference you have to find it out how varicella is different from smallpox. So if something happens like this, you can assure yourself that this is not smallpox because we don't have smallpox anymore. So those are actually varicella viruses, which is herpes virus. Okay, so this is the rash distribution, and based on that you can differentiate because smallpox is mostly on the face and the legs and the extremities, right? But your chickenpox is mostly in the upper body. Lower body you don't see too much. So that's the major to differentiate smallpox. But we don't want smallpox come back anymore. But the varicella is very characteristic. If you see the upper part of the body has the rashes, that's mostly varicella, even for adults and children. So.
Why is that virus there? Why does the virus?
There are two different viruses, and why I don't know. Definitely, they have different immune system functioning and how they affect the dermis layers, and yeah, I don't know. I think that's very valuable questions. I don't know how this happens. But definitely there are two different viruses. Their replication is little different. Their protein structure is totally different. I think the smallpox has more than 100 proteins, and herpes also is a big virus, but not that big like pox. But they have also different. I think the virus itself, the structural proteins and non-structural is totally different from the pox. I think that's major because they are two different viruses. But their symptoms may be similar. They both causes rashes, but the where it's exposed, like a West Nile, right? We discussed yesterday. West Nile also causes rashes, but it's only on the chest area. So now question comes, oh, why is West Nile not in leg? I mean, it's the virus, how they replicate. Maybe the virus is more prone to that area, regions. I don't know why it's not going to leg. It's a good question, but I don't know why. It says the virus signatures there, so that's the way. I mean, this is good another way, like we can differentiate easily. But if it's everywhere, like smallpox, it's a hard time to differentiate, oh, what virus it is. So I think it's nature's creation. I think virus is different. I think that's the way. Yeah, I mean, we need more studies on those. I mean, definitely, I think my major answer is those are two different viruses. and they have two different proteins. And even though people are doing so much research on how the virus host cell interaction happens, which protein is actually responsible for doing the lysis. So those are, those are the studies. People have shown some, we cannot do post-box research anymore. To do the study, you need a very high security lab like the L04 lab to do those. But it needs government permission and all those, because we make sure those we don't leak those virus and cause pandemic again. So you cannot do a smallpox research anymore. You need a high authority permission. Yeah. Yeah, some lab has. Yeah, I think Russia has, USA has, Europe has. Yeah. They still maintain. They are making that's why I mentioned, like they make like USA, I know better. They have that's why they are making vaccines for the army people, just in case there is any bioterrorism or anything. They can treat those army people first, and then if that's needed, then they go give to the civilians. But the priority is to the army people and the frontier health workers, like the doctors or the others who goes to the field and war region and those things. For them, it's important. No. Smallpox is eradicated. It's closed chapter. Yeah, it's closed chapter. We don't want it again. I mean, you saw the pictures, right? It's a horrible. So the 10th century Chinese inhaled powder scabs. So this is the immunization history that that's we discussed, like 1721, the subcutaneous inclusion scab tried during an outbreak in Boston, variola. We talked about cowpox yesterday with some people here. 1796, Edward Jenner used the cowpox scabs and used the vaccine. And 1967, WHO began smallpox eradication effort, which is vaccinia. Uh, so this is a good question. The evidence that general source of cowpox was not cowpox. So is that horse and rod? We don't know. Okay, but based on the evidence, they have shown that Edward Jenner is the cowpox vaccine for controlling. So vaccinia in Brazil, 1804, importations of recently vaccinated and enslaved people, standardized practice to vaccinate arm to arm, and that's all the kind of history here. Unclear if those strains are vaccinia gone wild or originating from the same. So these are the kind of questions still there. How, because there is not too much report there, right? Like here, right now, but that time Edward Jenner said, oh, it is cowpox. Everyone too has to believe this, and whatever reported Edward Jenner did. So those are the questions. So these are the kind of needles they used. So the bifurcated vaccination needles. I think so many people here don't know those. So you take those needles and you have the vaccinia, and then you just put in the dermis and make a scar there. So that way the vaccine haven't given to them. So that makes some injury to the skins and make a scar there, and that's why the vaccinate happens. Huh? The substance is here. This is the needle. Here you see the vials here. So you just reconstitute the vial and take that, dip into it, and scab it on its skin, make a scar, so that it goes to the dermis layer, and then the virus will be, going, taken by macrophages and everything. That, mostly the Langerhans cells present in the skin, so they take those present antigens to the T cell, and then go T helper cells, and then B cells take it, neutralizing antibody, and then go on. So when you do the vaccination, it's after day 6, you can see that kind of mark. So if you see that kind of mark, that is good thing. That means vaccination is happening. It's good reaction. And also it has been, so you see the level 2002, you know, and you see, and then day 7 it is increased, and day 15 it is like this. So that means the vaccination is happening properly. Your body immune system is functioning well. But every vaccination has some adverse effect, right? So these vaccines are also, because This is a live attenuated vaccine. So if you have immunosuppression, you have the dermatitis, it may flare up the reaction, bad reactions. Uh, Also, like lower among people who have been previously vaccinated, then they may have adverse effect. Some arm pain, swelling. I think Have you taken flu vaccine? Anybody here? Flu vaccine? Nobody take flu vaccine? It's not mandatory. Huh? What about adult? Okay, so United States is they always say take vaccines. Even my doctor says take. I took before I travel here. So our whole family takes every year, and we'll talk afternoon talk and talk about more flu vaccines. So the reason I said because flu vaccine also it's a intradermal. It's not like bifurcated needle. It's intradermal, but it also has a, like you can feel. Oh, I have sore arm. If somebody touch it, oh, it's so painful. So that means it's reacting. The vaccine is working. That means the body immune system is functioning. Macrophage is doing its work. T cell, B cell is doing its work because there is inflammation. If you have inflammation or pain, means the cytokine level is high. You got the antigen. If you don't feel it, have I got the vaccine at all? Or I got a water? Because, yeah, because it's a foreign antigen, right? When body see a foreign antigen, then innate immune system come first, right? NK cells, the cytokine, neutrophil, interferon alpha, those comes. So here also the same thing, like arm pain, swelling, those are common. Systematic reactions, you can get somebody fever, same thing, flu vaccine, you get fever. Some people get fever, doctor says take some paracetamol or something. So those are the commons, but this is kind of like generalized vaccinia. Inadvertent, how many people out of 10,000, like 10,000, 10 to the power 6 got that kind of, adverse effect, that kind of report there. Um, so this is some kind of adverse effect. You can see sometimes, oops, yeah, some adverse effect you can see. There's swelling and and the eyes regions, and sometimes in the hand, but it is very rare. Some of the vaccines is always like this, but the federal vaccine have fewer than 50 cases so far reported. So many millions of people, billions people taking the vaccines, but some few cases, even for measles also, right? Their adverse effect happens. So this is the same case. Myopericarditis, some cardiac dysfunction happens if you take that vaccine, but it's very rare. Relatively long incubation period, 14 days, low infection rate. So, and a good thing is uh, the rate of transmission. Okay, rate of transmission is very poor. It's like I mentioned, like you have it mostly happens when it's hemorrhagic, right? Then it's very transmit easily. But the transmission from one person, if I have a smallpox, I to give Dr. Reddy or Dr. Ashok, it's very low because the value of R is low. So the more is measles. Measles is very high. Its RO is 10. Like if I have measles, I can easily because it goes aerosol. COVID is very high transmission. If I sneeze, there are more chance I can transmit others. But the smallpox is very low, which is good. And a case of infection was one for lack of extraction reservoirs. This did the mass vaccination that time. So this is the dedication. Yeah, I told right, 1975. First time happened in India, 1975, and Europe happened earlier, 1953. Well, Europe is the developed country, right? Developing country always late. 1975. Even Africa is late, 1977. And so this is a painting displaying the panel based on recognition card, like promising a reward. So that time they have a reward thing, like if somebody find out a people has a like smallpox vaccination not done, they'll give you some money so that if you can recognize. So that because the idea is to get everybody vaccinated so that the disease can be cured. So this is the poster in Bangladesh, 1975. They are putting that anybody knows this, that kind of symptoms, just let us know. We give you vaccine. So now going to mpox. So this is earlier called monkeypox. This is the same family, orthopox virus, and the same poxviridae. And what the virus looks like this, it's a circular, okay? The smallpox was like a brick shape. Uh, so the pox virus history is long, 1958, and it's mostly detected in monkeys. Then the first outbreak happened in Democratic Republic of Congo in Africa, 1970s. And then it's very like sometimes it's there, sometimes not. Like some cases, like 6 cases, 71 localized, and then USA first outbreak 2003. So you can read those slides easily. I want to focus on 2022. So that is the major thing happens like 2 years after the COVID. Probably you have heard that the monkeypox, right? The monkeypox is 2022. The largest multi-country outbreak happens, and WHO declared it's a public health emerging infectious disease. They make it a committee. There are 99,000 cases, 207 deaths, and 122 countries infected. Most cases in non-endemic countries and rapidly spread by sexual contact. So this is mostly like HIV is transmitted through sexual transmissions, mostly like gay people, homosexual people. Um, and then official end, but they have a vaccine, so they're able to control. I think major is the sexual contact. So if you just control that part, and then they have a vaccine. So it has been 2023 officially they closed, so the outbreak stopped. So they take very quick measures of those, and it stopped there. But there are some outbreaks occasionally, mostly found out, if you see the newspaper and other, look over world news, there's still some outbreak happening in different African countries, and it's still there. So, but there is a good vaccine already available. So here is some of the cases what is reported, and you can see a lot of countries, especially United States, has a highest number, and you can see China, Portugal, Italy. I don't see India here, right? But you can see there is India. There are some outbreaks which is not in that figure, very lower figure, is been reported. So it is very globally. It's a pandemic. So globally, there's a distribution is there. The countries that have historically reported monkeypox was in Africa, and still there's some reports there. And between 22, 23, that is the countries were reported. So these are the countries, as I said, still now there are some sporadic outbreak happening in Africa. Yeah, I said, India is lower than 100. So there's one death in India has been found out in 2022 due to the mpox infections. So this is the map of Africa reported. We are somewhat interested in this, and in this just to show how what is happening in different countries like Nigeria, Liberia, and those. You can see more happen in this dark red area, which is Republic of Congo, which is also close to also the Democratic Republic of Congo. This is the country where tomorrow I'll talk about a lot of Ebola infection also there. So this is the countries has everything kind of disease happens that time. So and it's the monthly cases how it's reported, those are there. So here it's showing like 2024 each year and like 2022. So that's sporadic infections mostly happening in Democratic Republic Congo's in African country. Yeah, and it's mostly transmitted through sexual transmission. So how they are protecting and all those things, some of them they don't have vaccine either because they are poor country, right? Right, right. So we are not too much interested in phylogeny, but but you can see there are a lot of clade separations here. There is Sierra Leone, which is one group, is a big cluster, and then Gabon. So different. This is the different parts of African country, and they did the phylogenetic tree of the by the next generation sequencing analysis of this mpox, and then they cluster those viruses. The idea of this kind of, you can see a lot of papers has a phylogenetic tree. The idea is to find out where the transmission happens, right? Is that because if you see, like, I take that Indian, which is one died, my mpox, I collect samples from that patients, or the died patients, and then isolate the virus, okay, and then match with the USA or Africa or everywhere, right? Get those viruses isolates and match with them, and want to see how they fall. What is the relationship those? Okay, and if you see they are distant relationship, so this is like a West Africa and Central. I think it's the Central East. I forgot what it is. So West Africa and this area has a different place. So they are separate. Those viruses are not come from the same place. But within that place, like if you see like West Africa, like Sion, Liberia, and then Paris, the Paris strain, Copenhagen, Europe, they are very close. So we can say, okay, the virus came maybe from Africa or Africa to others. They are very close. So that the transmission might happen somehow, maybe the traveling or maybe other methods. Okay, so that's the idea of the phylogenetic tree here. Yeah, West African clades, but they found out no mortality rate, no human to human transmissions here. So the same thing here, they did so many phylogenetic tree to see what is the relationship between those viruses. So clinical disease is very similar to discrete non-complicated smallpox plus lymphadenopathy. Uh, Fever, headache, back pain, those things are there. 1 to 3 days fever, oropharyngeal lesions, so it's a pharynx in the throat, upper respiratory involvement, illness lasts for 2 to 4 years. So CFR means case fatality rate. Only fatality is 9 to 8%. Unvaccinated children less than 8 years. So these are the vaccinated, and this is vaccinated and unvaccinated. The comparison is this: if you are vaccinated, then fever to rash onset is very longer. Lymphadenopathy reduced. Other symptoms you reduced, right? If you are vaccinated, but you get the symptoms, but the vaccine, that you'll not be dead, or your symptoms will reduce. So this is the symptoms, like same like smallpox. They rash. They also do the same kind of things, like pre-eruption phase. You can see the rashes in the body, in the chest. The day 4 and day 5, their scabbing started, right? They are drying. Those pustules can dry and all those happening. The same thing, the rashes are similar like smallpox. You can see those lesions everywhere. You cannot differentiate between smallpox and monkeypox. It's kind of similar. Looks like the baby has eruptions stage and some part scabbing happening, and you can see those marks. So these marks will not go away. It's kind of permanent. You can see those babies here. Yeah, this is case recovering well. So here that guy is recovering, and you can see the lesions are healing. They're no more like pustule form. The back side also is showing some pictures showing this. So there is a studies has been done in showing that there are so many squirrels. Okay, they are actually natural. They are kind of harboring those viruses in them, because in Africa they try to feed them, they'll try to create them, and they play with them, and maybe they got the virus from them. So this is a study. There are some publication on that. Uh, those squirrels are also, there are different species present, and it's very prevalent in the African regions, and they have a huge human to contact. So they are feeding that squirrel, and somehow in the saliva, or urine, or fecal matters they get contaminated, and there may be chance to get the humans that infected.
So,
So this like this, huh?
it is also one of the major prevalent in monkeys.
What?
Is this virus prevalent in monkeys?
Yeah, yeah. That's why the Africa is prevalent. Yeah. So this is the cycle within animals also it circulates, but nobody thinks about how much virus they have, but it circulates. They don't die too much. That does not impact too much. But it got contact to infected animals as well as humans may get uh, exposed to when they are feeding them, or just petting those monkeys, or having I have a pet rat, or those things, so they get infected. A lot of tissues got and organs got involved in it. It may got CNS, encephalitis. The virus goes to the brain too. Eyes, oral, lymph node, cardiac, and so name it, they have it. Even urogenital systems, skins, definitely. That's why the rashes, digestive system. So all kind of organs they can get involved with the mpox. So it's a kind of systemic infection. It's not like a localized infection, like pox. And that is the all the symptoms say here is listed. I mean, it's nothing usual here like smallpox. The differences. Another important is there is a chance of vertical transmission as we know. One is contact transmission, which is mostly happening through sexual intercourse. Social transmission also happens as a horizontal transmission. And another is vertical transmission happens if the mother is infected, the baby can get as mpox. There are a lot of cells involved. Once it binds to the receptors, that those triggers all kind of antigen presenting cells. So there's a macrophage taken by this, and then by the phagocytosis, monocytes and neutrophil also comes in because neutrophil is one of the major also for innate function. Innate means it's come always first, and then those are the adaptive immune response which come later. CD4 cells, which is a helper, CD8, B cells, and all those. And you can see CD4 is a T helper 1, T helper 2. It's like a based on what cytokine they produce, they have to define T helper 1, T helper 2, and interferon gamma. There is also CD8 also produces perforin because it's a killing. You have to kill those cells, infected cells, so they increase those. B cell produces a lot of antibody. ADCC means antibody dependent cytotoxicity. So B cell is involved. B cell produces antibody, and then it binds to the virus, and then the macrophage kill it. So it's a called antigen dependent cytotoxicity, or CD8 function only when the virus is bind to the antibody. NK cells, there are a lot of papers coming. We know the NK cells is an innate function. NK means natural killer cells. As its name says, natural killer means it has no specific things to target. Any viruses comes, natural killer cells try to react on those. They increase again the proliferation. Due to proliferation, the count increase, but their effector function decrease, the chemokine decrease because they are not completely protect those things. And there's the activation of complement, which is very important for get rid of those infections. So this is a cascade. I mean, most of the viruses do the same thing. Antigen presentation is very happening. I think tomorrow I can discuss about how the antigen presentation happens. So some videos on it. And differentiate between innate and adaptive immune response, so you can have some clear idea of what we are talking about. So you can note it down and we can discuss. So this is the stage of skin lesions. Macule, Dr. Marty was asking about like what are those stages are. These are the pictures. So the macules is like this, which is not well formed yet. And then papules, you can see there's a raised on all those. And vesicles means it's full of fluids. And the pustules means it has a pus formations. And it looks like white. And the scabs, when they burst, and then you can see those hemorrhages and all those things. So this is the typical lesions happen for pox as well as mpox. Control, okay, it has a good control because the vaccine. the MVA, vaccinia vector. Okay, MVA vaccines, they use a lot of vaccines presents uh, that controls those mpox vaccines. So it is a vaccine available. Avoid sex and social gathering. That is the preventive measures. Avoid close and skin-to-skin contact with people who have a rash that looks like mpox. Try to cover, try to not to touch. Avoid contact with animals where mpox occurs. Avoid contact with objects, material that a person with mpox has used. Wash your hands often. That's some of the, except the first one, all this I think we can control, right? Without any paying a single paisa, right? Simple. Wash your hands, be careful. So I think it's kind of a simple strategy we can follow, not only for mpox, for all the diseases, right? Mostly the transmission happens, as we said, like horizontal. That's more we care about. Like vertical transmission, it's only girls or women should take care of it. But they should be screened before, like before pregnancy. They don't have any other viruses. We work a lot like hepatitis, herpes simplex virus 1 and 2. This is very common. I don't know. You ladies take the HSV1 vaccine or no? I don't know India, but my daughter takes. This is a United States. I think they have a HSV1 and 2 vaccines. That also protects. This is herpes simplex virus and also papillomavirus. Those are another, which is an oncogenic virus. So there is vaccines available. So that also gives protection in the sense when the lady got pregnant, she should not transmit to the there's less chance of vertical transmission of the virus, so the baby will be healthy. Um, but all those things is easily can be taken care of by general knowledge, I should say, and that way we can control the mpox here. I think that's the last slide, I guess. Yes.
US strictly follows these things, right?
What?
US follows strictly.
Yeah, yeah. That's why.
It is prevalent there.
Mpox.
And
Well,
also, they don't play like us,
so yeah, so mpox was very prevalent in United States, but if you think about how much it's a different thing. It's a culture thing. Okay, United States has a lot of homosexuality, and that if you read this, that all the mpox happen men sex with men, that kind of. It's mostly prevalent in men sex with men, and it is only prevalent in between homosexual persons. They are very rare in the heterosexual people. And if you go to California or even New York, one out of I shouldn't say one out of, at least 2 or 3 out of 10 people are homosexual. So yeah, 1930s it was mostly Africa, right? It was mostly Africa region, and then it came, it transmitted to the human because it's the spillover happened, right? Well, virus always spillover happens from animal because we don't know how much virus is there in the animals. So many. And when it spill over to the human, then we are very concerned. Oh, we have to cure it, we have to do something, make vaccines, all those things.
but they usually distance from these animals. That's what I think.
Yeah, it's the same like HIV camps, right? The theory says HIV came from monkeys, but it came to human. Now the virus has adapted to the human, so it is not going back to animal again. And the transmission is happening, and we don't know how the transmission is happening. Then it is bad, right? Now we know the impacts is most transmission happen to the sexual contact, but earlier people don't know that much. And that's why the transmission, same like HIV. So, intravenous drugs is like a lot of people taking drugs with a syringe and all those. That's also contaminated. Like I'm using the same syringe, other people are using the same syringe. So that's another cross contamination. Like COVID. Why COVID? Because COVID is not new. COVID happened long ago, and then MARS came, then COVID-19 came. But we know COVID is a respiratory infection. But even though, nobody was wearing mask, and the virus already transmitted so many million of people, right? And then when it is already there, then we are wearing mask, and but it's too late, right? So that's another thing. Even like United States, at some point, they say mask is only mandatory for those healthcare peoples. So general people stop using those. But they'll later say, oh, everybody should wear masks. I think because the knowledge is coming, and then people digesting the knowledge and say, oh, we need to do this or that way. So that's happening. So we are continuously learning those things, uh, but, but it is there. I mean, despite all this knowledge, even the developing country, you're right. Like they are always taking precautions, and they are not too much population like other countries, but still they have it. So yeah, because they have other behavioral issues, they have other things which is actually um, taking a role in transmission.
Mandatory, they made mandatory to take all the vaccines.
Yeah, they have vaccine. Now they don't have any. Yeah.
Only 48.
Yeah. Yeah. So it is better now. Yeah. Any questions? Yes.
I'm not very sure about it, but from what I have read, lymph nodes are sort of the meeting point for a lot of immune cells.
Right.
So when you say HIV and some other diseases stay there dormant, how is it that they're not recognized by these cells, or and most that that's where they're sitting, where the cells are?
So HIV is a separate, different kind of very complicated virus. So it has a enzyme which is called reverse transcriptase and integrase. Okay, those two enzymes are very important. The virus made that. So what is integrase does? Integrase it integrates like my host RNA or DNA is there. So my host DNA is there. It made integrase, and it made DNA by reverse transcriptase, and it bind to the host, and then it stay there for forever, and it does not do anything. So the virus is present, even though if you give anything. So the drugs, if you give the drugs mostly against like the capsid proteins or reverse transcript, but there is no reverse transcript. It's already integrated with the virus, the host DNA, so it stay there. Now, what the virus, another mechanism it has, like once it says, okay, my immune system is not strong enough, the virus start multiplying again. So it has a DNA, it has double-stranded RNA. So the RNA tries, the positive strands tries come out because it has a cleavage enzyme. So it cleaves from the host DNA, it starts to mRNA and generates their own protein again. And that's the unique for HIV. That's why it is very difficult to eradicate HIV. And then it starts multiplying, it makes virus and produce so much. It's not only lymph node, it's present in the brain, it's present in the gut. There are a lot of, we have studied in the colon, in jejunum, different organs. It lives in the tonsils, it's lived in the kidney, liver, so many places. So yeah, they stay there. And what are those cells? They are some we found is mostly the naive cells. So we can talk later on. There are people define different cells like T cells, like naive T cells, activated T cells, proliferating T cells, memory T cells. So they have different terminology, but you can get an idea like why naive. Naive means what? If I talk
Probably have not had interaction.
yeah, naive means naive. Like if somebody said, "Do you know SP Balasubramanian?" No. That means I'm naive that he is a good singer, right? So that means I'm naive. So some T cells does not know. They have no memory. They don't know. They don't see any antigen. So they are naive. So those naive cells are getting infected. Those HIV, and they keep those viruses there. Mostly naive cells. But the virus mostly infect the activated cells. And then activated cells, when they replicate, they multiply, the naive cells coming, so the naive cells also present their inside, and they stay there. some we also saw the memory cells also present. So it's not like only one cell so naive or memory. Everywhere it is there, but we still don't know what is the mechanism happening. But the point is like they have the integrase, they have the reverse transcript. That's the enzyme. They actually play a role to stay there for longer. The immune system cannot do. Even the lymph node, there are a lot of follicular T cells coming, macrophage coming. They cannot do anything to cure.
So could it be that these cells are getting infected elsewhere, and since they're immune cells, they are bound to go to the lymph node and hence they settle there? Is that how that works?
Of course, yeah. So that's why you got the virus, all the virus like when you talk, right? They go to lymph node because the lymph node is the major. So it's kind of a factory house, like you got something. It's like a screening, like when you go to airport, like they tell they scan you. Do you have any bomb or rifle or any poison or any powder? They scan you or metals. You are not taking any firearms, knife or something. So the screening, right? So lymph node is some kind of organ. They screen. So every blood is going lymph node. The lymph node's job is, oh, I have to find out anything bad things coming or not, and they screen it. And then they find out, oh, something wrong, some foreign things came. Then macrophage come in. Macrophage come in, engulf it. It also happens in the peripheral blood, but mostly happen in the tissue. The circulatory macrophage is less. It has circulatory macrophage, but mostly macrophage are present in the tissues and all this. So the tissue's function is those. The tissue macrophage, lymph node macrophage, they grab those bad things, like foreign things, and then they try to kill those. But they cannot kill mostly for HIV. Yeah. But for like Chandripura virus, Sudok, like dengue, they can do easily. But HIV is impossible. The HIV we studied and others did a lot of studies. HIV stay as a reservoir. We talk human papillomavirus, HSV1 and 2, herpes simplex virus. It's very common in human populations, but it does not do too much bad things. It is very mild kind of. If you tackle it, you're okay. I have a HSV1, okay, but I'm not going to die. But the HIV is bad. They can take care, they control other cells, like especially CD4 cells. And I say CD4 is a helper cell. So if the helper is bad, you ruin your whole system, your functional system. So, yeah, HIV is a different story. It's deviates from all the other viral mechanisms. So.
All right, if there are no